Capture theory of the universe. Refuted. Some people out there have some ideas about how the planets formed and so on. The theory. Planets and moons were flying around and some were captured by our sun and began circling. Why the theory is not true. Outer space is too large for 9 planets and 60 moons to be caught by our sun. Millions would have to pass in order for one to be caught. No planets or moons are flying around by us today. They would tend to crash into the sun, not fly by it, or begin encircling it. Moons would not begin orbiting around planets. They would crash into the sun, or into other planets. The accretion theory. The theory of pile of space dust and rock chunks pushed together into our planet, and another pile pushed itself into our moon. Then the moon got close enough and began circling the Earth. Why the theory is not true. Where did the space dust and rock come from? Loose gravel, etc. in outer space would not push itself together, it would push apart. The moons and planets would crash together. Planetary collision theory. The theory, our world collided with a small planet, and the explosion threw off rocks, which became the moon, and then it began orbiting us. Why the theory is not true? Such an impact would destroy the Earth. Material from the explosion would keep moving outward forever. Outward moving material would not stop and begin circling. Such an event would have to happen to all the other planets. Thousands of near misses would have to occur for one to crash together, yet no moons are passing by us today. Stellar collision theory. The theory. Our planets, moons, and suns spun off from the collision between stars. Why the theory is not true? The collision would hurl outward material outward, and never veer from that outward course. Any pieces drawn together would smash, not orbit one another. Gas cloud theory. The theory. Gas clouds were captured by our sun, but instead of being drawn into it, they began whirling and pushing themselves into planets and moons. Why the theory is not true? Gas does not lump together, it only spreads outward. If gas could stick together, it would not produce objects which would encircle the sun, nor would smaller bodies encircle them. Chemical evolution theory. David Rossiter, director of Creation Science Movement in England. The myth of chemical evolution. The ancient Greeks believed in the spontaneous generation of life. More recently, Louis Pasteur showed that life did not arise from non-living material. Yet those who deny the Creator's existence must believe it. It happened once upon a time. Evolutionists estimate the Earth to be 4.6 billion years old, and the earliest fossils are about 3.8 billion years old. An initially hot Earth might take, say, 0.3 billion years to become user-friendly, so the first life took only about half a billion years to arrive from abiotic non-living, starting materials. If it is as easy as just having the right conditions, one might think that life should have evolved many times before the advent of photosynthesis produced an oxygen concentration which made conditions unfavorable. Yet all life rides upon the same biomolecules, metabolic pathways, and genetic information. So life had but one origin, either created or evolved. Modern theories of the origin of life date back to the Soviet scientist Oparin in 1924. His ideas of a primeval soup were promoted in the West by fellow communist J.D.S. Haldane of Cambridge. In 1953, Urey and Miller published results of some simple experiments in organic chemistry which seem to lend credence to the soup theory. Interestingly, 40 years later, Miller admitted that the question of the origin of life is much more difficult than he or anyone else had thought. Clutching at straws, others have suggested mid-ocean ranges with their cocktail of hot chemicals as the cradle of life, while others have postulated an extraterrestrial seeding of the Earth. This later suggestion still does not offer a mechanism for abiogenesis. Now, with the development of molecular biology, since the time of Oparin and Haldane, the cell is no longer regarded as simple. The living plasma membrane shows in or out only specific compounds. It is not simply a semi-permeable membrane. Cells contain nucleic acids that carry information about the structure and functions of the organism. They also contain ribosomes, where proteins are made using a complex mechanism of nucleic acids and more than 100 different proteins, each with a specific task. The cell also contains mitochondria, where energy, ATP, is produced. The complexity of all these parts of the cell is enormous. Lynn Margulis has suggested that the first protocell assimilated these organelles by a process of symbiosis. However, these components cannot now exist independently, nor could the cell exist without their combinations. Moreover, one such type of organelle, known as a lyosome, contains enzymes whose function is to digest foreign bodies. With all the amazing complex, mutually dependent components, it seems that the cell had to be complete from the beginning, rather than assembled piecemeal over years of evolution. The major biochemicals in living cells are proteins and nucleic acids. No biologically significant proteins or nucleic acids have been made by any experiments such as those of Miller or those who have followed him. Proteins are strings of amino acids whose enzymatic activities arise from active groups within a specific three-dimensional shape. These are due to a precise sequence of the amino acids. There are 20 amino acids found in proteins, although many others exist that are not used in metabolic pathways. Chemically, they are NH2CHRCOOH, where R is H in the simplest case, glycine, but can also be a variety of organic groups such as CH3 in alanine. In all amino acids except glycine, the central carbon is surrounded by a tetrahedron of 4 different groups: HR, amine, and carboxylic acid. Because of this asymmetry, amino acids exist in 2 forms, a designated right and left-handed, or D and L, or R and S. In proteins, a string of amino acids, NHCHR, CONHCHR prime, CO, etc., formed by condensing out of a molecule of water, HOH, between each pair. All amino acids are left-handed. This left-handedness gives that chain of spiral twist leading to a specific 3D shape that is essential to its function. In the living cell, proteins are made by means of RNA and many specialized proteins or enzymes. Since proteins are needed to make proteins, it is not easy to speculate how a first protein could occur by chance processes. More and more, looking at a creative designer. In laboratory experiments aimed at simulating conditions on a lifeless Earth, a messy mixture of amino acids can be formed, consisting of mostly glycine and D-lanolene. Not all amino acids found in proteins can be synthesized in this manner, while many not used in living systems do result from these experiments. a product consisting of exclusively left-handed amino acids never results, and from the theoretical considerations cannot result. Only D/L race mixtures are formed. Peptidization, the joining of the amino acids to form a protein by the elimination of water, is difficult to accomplish by non-biological means. Proteinoids are unstable in the presence of water. Since they cannot replicate themselves, natural selection cannot be a driving force in their improvement. The precise order of amino acids in proteins in cells is governed by information on the nucleic acids that code for them, so this could not be achieved by chance. Moreover, the tar-like byproducts would tend to poison any enzymatic activity in proteins. So nucleic acids are found, Nucleic acids are found in living cells as DNA, a ribosomal RNA, a messenger RNA, and transfer RNA, each with specific properties. What a complex design. They consist of strings of nucleotides, which are composed of a nitrogenous base, a sugar, and a phosphate linkage. DNA carries the genetic information for the organism, while RNA is used in protein synthesis. There are 4 different bases on the double-stranded DNA. The strands are linked by weak hydrogen bonds between bases on each strand. The structure of the bases is such that each in one strand only links with one other type of base in the other strand. One single strand of DNA therefore acts as a template for the other strand during cell replication. 3 consecutive bases act as a codon to transfer information to specify of a particular amino acid, or to start or stop a sequence. This information of a string of DNA gene is responsible for the formation of a particular protein. Since there are 4 bases, there are 64 codons, 4 to the 3rd power, which pass information rather like the letters and punctuation of a written message. This is a precise mechanism. Information transfer is checked at a rapid rate by proteins for random changes known as mutations, which lose information. No mutation could lead to an increase of information, so neo-Darwinism cannot be a mechanism for macroevolution. It is a tenet of information theory that information only comes from an intelligent source. So genetic information must have been created. Information not only implies meaning, but purpose. This is the opposite of chance. As a carrier of information, the DNA molecule is 4.5 to the 10th to the 13th power, 45 trillion times more efficient than the silicon megachip, which was made by teams of designers. Incidentally, a stereochemical basis for the relationship between any particular 3 nucleotides and the amino acid for which they code has not yet been elucidated. When nucleotides are joined in a laboratory, thermodynamic considerations allow one particular site for bonding phosphate to sugar to the next phosphate. However, such a pseudo-DNA strand is not biologically useful. The bonding site found in DNA, the 3' and 5' carbons of ribose, is in the best position because the proteins used to join it together acts as templates to get the junction across the sugar right. The sugars, deoxyribose in DNA and ribose in RNA, are also chiral like the amino acids, but in this case the sugars are all right-handed. Again, there is no conceivable mechanism for arranging this by chance.